Good morning, my dear students. I am Shivaram Krishna, lecturer in Botany, Government Degree College, Puttur. Now I am going to present a topic on plant growth and development in the fourth semester from the plant physiology from the fifth unit, plant growth and development. The subtopic is phytohormones, auxins, and gibberellins. Generally, we know about the hormones. Generally, hormones are useful for the growth and development. But what are phytohormones? Phytohormones means in the name itself, phyto means plants, hormones means generally they are the internal growth regulators. Phytohormones are the organic compounds produced by the plants for the purpose of growth and development. Next, what is the hormone? Generally, hormone is a chemical substance produced in very low quantities for coordinate to the process of the growth and development. Generally, hormones they are synthesized in one part and show its effect on other parts by its transportation. Next, phytohormones generally called as plant hormones by the some physiologists are plant growth regulators. There are the five major group of plant hormones are there. That is the auxins, gibberellins, cytokinins, abscisic acid, and ethylene. These are the five major classes of phytohormones. And brassinosteroids, jasmonic acid, salicylic acid are also under the category of phytohormones. Generally, phytohormones are divided into two types. One is the plant growth promoters and the plant inhibitors. From the plant growth promoters category, auxins, gibberellins, and the cytokinins. Generally, these are the plant growth promoter category, and inhibitors, abscisic acid and ethylene. These are the inhibitor category. Next, first phytohormone auxin. Generally, auxin is a Greek word. It means to increase or grow. The discovery of auxins were the first plant growth hormones to be discovered. Charles Darwin and his son Francis Darwin observed that the tip of the coleoptile, that coleoptile means the protective sheath in the canary grass, it grows and bends towards the source of the light. When grown canary grass in the dark, it grows straight but bent towards the light. Generally, the bent of the plant towards the light, the phenomenon is called as phototropism. Then, after the Darwin's experiment, next Boyson and Jensen in 1913, they conduct the experiments by decapitation of the coleoptile, and the decapitated coleoptile placed on the agar block and revealed the substance that the coleoptile, the substance is diffused from the coleoptile stump into the agar block. When the agar block is placed on the plant, it causes the differential growth. Later, after the Boyson and Jensen in 1928, the Dutch botanist F.W. Wendt, he isolated from the auxins from the tip of the oat coleoptiles. Following the Wendt's success, auxin, that is indole acetic acid IAH, it is isolated from the human urine, then from fungi and other plants. Generally, we can whatever we earlier the Darwin experiments, Boyce and Jensen experiments, then Paul, then later by the Wendt. This we can understand through these figures. First, Darwin experiments. Darwin he conducted that first he grown the plant within the light. He observed that coleoptile bending. Then the coleoptile stump is removed. There is no bending is occurred. Then he placed some cap on the coleoptile stump. There is no bending is observed. Next, Boyce and Jensen, the tip is removed in the canary grass. Next, he placed gelatin between that decapitated part. Then he observed that again the bending towards the light is observed. From this Boyce and Jensen experiment, he noticed that some substance from the coleoptile tip is diffused towards the stump portion, from the coleoptile tip towards the stump portion. He observed by these things observed by the Boyce and Jensen. Next, in the Paul experiments, he also removed same experiments conducted by the Boyce and Jensen, but he removed the tip, and the tip is placed on one side of the stump. Then he observed that the portion where the tip is located, that part only showing the bending towards the light. From this, what he understand, the substance is diffused and cause the differential bending. Then in the Wendt experiments, Wendt he decapitated the coleoptile tips and placed on the agar blocks. Then the agar are made into small blocks by cutting. Then the agar blocks, the cutted agar portions, they are placed on the stump part, the decapitated stump part. Then it also shows the bending. From these experiments, the scientists concluded that the substance is chemical in nature and it is diffused from the coleoptile towards the stump and cause the differential bending. Then synthesis. Generally, auxins for its synthesis, it requires the tryptophan, which is an amino acid. It is a precursor for the synthesis of auxins. Generally, auxins are most actively synthesized in young leaves, shoot apical meristem, flowers, and developing fruits, embryo, and the pollen. Generally, the main source of auxin is generally shoot apical meristem. Transport. After its synthesis, it transports. The auxin transport is polar. That means always in one direction. Generally, the main source of auxin synthesis is shoot apical meristem. From the shoot tip, it is transported towards the other parts through the phloem tissue. Next, auxin types. Generally, there are naturally occurring auxins and the synthetic auxins. Both forms are available. Naturally occurring auxins are IAA, IBA, and IPA. IAA means indole triacetic acid. IBA means indole 3-butyric acid. IPA means indole propionic acid. Then synthetic forms of auxins are also available commercially, that is NAA and 2,4-D. NAA means naphthalene acetic acid. Next 2,4-D means 2,4-dichlorophenolic acetic acid. Next, these are the chemical structures of IAA, IBA, NPA, IBA, and the 2,4-D. These are the chemical structures which it represents the indole ring. Next, physiological role of auxins. Generally, there are different types of the physiological responses we can observe through the effect of auxins. One is cellular elongation, cell differentiation, root initiation, apical dominance, prevents abscission. Next, flower formation, parthenographic fruit development as a weed killer, and sex expression. First, cellular elongation. Generally, auxins elongate the cell wall by increasing the cell wall plasticity. The acid growth hypothesis explains how IAA increases the cell wall plasticity. This acid growth hypothesis, generally first IAA stimulates the H plus pumps in the cell membrane, then H plus pumps secrete the H plus into the cell wall. The increased H plus concentration decreases its pH, that means acidifies. This acidified cell wall which activates the pH dependent enzymes and break the bonds between the cellulose microfibrils. As a result, the cell wall becomes loosened, and the turgor also helps to expand the cell. Next, cell differentiation. Generally, cell differentiation is a morphological process. Morphological differentiation occurs. Generally, auxin and sugars in its combinations required for the vascular tissue formation in the tissue culture studies, and auxin and low sugar concentration results in the formation of the xylem, and auxins and high sugar concentration, that means up to 4%, it is required for the phloem differentiation, and auxin and moderate levels of sugar, that means 2.5 to 3.3%, it differentiates the xylem and phloem. That means what we understand, the cell differentiation is based upon the auxin and the sugar. Both will influence the cell differentiation process, but its concentration is very important in the formation of vascular tissues like the xylem and phloem. Next, root initiation. Generally, auxins stimulate the formation of adventitious roots in the stem cuttings. That means in many nurseries, they are propagating the plants through the stem cuttings by using these auxins. The synthetic auxin Na is widely used as a rooting hormone. Generally, in the market, where there are so many synthetic auxin hormones are available, these nurseries generally they are practicing in the root initiation of many stem cuttings. Generally, apical dominance and inhibition of lateral buds. Apical dominance, that means apical parts exhibit the dominance. That means terminal or apical buds exhibit the inhibitory influence on the axillary buds and preventing their development. This phenomenon is known as apical dominance. Generally, auxins are produced at apical meristem and suppresses the development of axillary shoots. That means when the source at the shoot apical meristem is removed, but it removes the apical dominance. When the shoot apical meristem is present, it exhibits the inhibition of lateral buds. This is called apical dominance. It is the one of the physiological effect of the auxin hormone. Next, prevents the abscission. Abscission means falling from the plant. Generally, the leaves as well as the flowers, fruits generally fall from the plant early. That is called as abscission. That the abscission layer is formed as a result. The integrity loses between the plant part and the plant. Then it falls from the plant. Generally, when the auxins are sprayed, it prevents the early falling of the leaves, flowers, and the fruits before its maturity. Next, flower formation. Generally, auxins generally inhibit the flowering, but in pineapple, auxin induces early flowering. Auxin also promote the development of flowers in winter barley. Next, parthenocarpic fruit development. Generally, parthenocarpic fruit means the fruit which does not have the seed. That means seedless fruits. Generally, in the market, we are observing the grapes and that other like the tomatoes and other citrus fruits without any seeds, and that watermelon also without seeds we are observing in the markets. Generally, the parthenocarpic fruit development is the result of the effect of auxins. First, Gustafsson he discovered that auxin could induce the parthenocarpy in the plants. The auxin treatment of young unpollinated ovaries in tomato, grapes, pears, and citrus fruits resulted the development of parthenocarpic fruits. And IBA is also induced the parthenocarpic fruits in some plants. The weed killers like the 2,4-D and 2,4-D and 2,4,5. That 2,4-D means 2,4-dichlorophenolic acid and 2,4,5-trichloroaxyphenolic acid are used as a weed killers. The weedicides are only effective against the broad-leaved weeds. That means synthetic auxins like the 2,4-D and 2,4,5 act as the weed killers. And sex expression finally. Generally, auxins can modify the sex of the flowers. The application of auxins produce the female flowers instead of male flowers in cucurbitaceae. Generally, cucurbitaceae there are the male flowers and female flowers are there, but female flowers are of importance because the fruit set is based on the female flower development of the female flowers. Then it is of commercial importance the development of the female flowers. When you apply the auxins externally, it leads to the development of more female flowers instead of the male flowers. These are the some physiological effects of the auxins. This is the figure of apical dominance where we can see the that active bud suppresses the lateral bud development. In the second figure, we can observe that when the apical stem is removed, the development of the lateral buds is observed. Next, this is the parthenocarpic fruit, watermelon, where there is no seeds. It is one of the physiological effect of the auxin hormone. Then coming to the gibberellins. The discovery of the gibberellins is Gibberellins discovered mainly in the Japan while studying a fungal disease in a rice called Buccaneer foolish seedling disease. That means Buccaneer foolish seedling disease means the plant grows very tall without any seed set. That is called as the Buccaneer foolish seedling disease. Generally, the disease is first observed in Japan. It is due to the fungal disease Gibberella fujikuroi. That fungus caused this Buccaneer foolish seedling disease. Next, Kurosawa, a Japanese physiologist, discovered the gibberellins are secreted from the Ascomystis fungus Gibberella fujikuroi, which cause the Buccaneer disease. Next, Yabuta and Haishi, they crystallized the chemical substance from the fungus and named it as gibberellin. More than 100 gibberellins identified so far, and 40 are reported from the plants and remaining from the fungal organisms. And the identified gibberellins are named as GA1, GA2, and GA3 based on the sequence of their discovery. Most intensively studied gibberellin is GA3. Next, chemical structure of the gibberellins. Gibberellins are chemically they are tetracyclic diterpenoid compounds which are consist of 4 isoprene units. Generally, chemical formula it is represented by C5H8. Next is synthesis and transport. Generally, gibberellin synthesis occurs in mainly young leaves and the buds and developing seeds, fruits, and in roots. Next, transport. Gibberellins transported passively through xylem and phloem. They transport by non-polar method. Earlier in the auxins, the transport is polar. For gibberellins, it is non-polar transport. That means in both directions they will move. Next, physiological role of gibberellins. Remove the genetic dwarfism, seed germination, internode elongation, bolting, flowering, substitutes the fertilization, parthenocarpic fruit development, sex determination, improve the fruit size and the shape. These are the physiological roles of gibberellins. First, removal of genetic dwarfism. When the genetically dwarfed plants increases its height when the exogenous application of gibberellins is applied. That means the dwarf plants can be improved its height by the exogenous application of the gibberellin. That is the effect of the gibberellins. It removes the genetical dwarfism also. Next, seed germination. Generally, gibberellins accelerate the seed germination by breaking the seed dormancy. In germinating seed, embryo secretes the gibberellic acid and induce the synthesis of amylase and other hydrolytic enzymes which help to mobilize the food reserves from the endosperm towards the embryo. Then it starts the germination. That's why gibberellins cause the seed germination. Next, stem elongation. In sugarcane, it is practicing exogenous application of gibberellins. It leads to the elongation of the internodes. When the elongation of internodes leads to the increase of the yield. In sugarcane, generally, if the sugarcane having very short nodes, there is no product yield. That's why when the application of the gibberellins to the sugarcane crop, it increases the internodes to an increase its length of the internodes. Then automatically the productivity increases, and ultimately the yield also improved. Next, bolting. Bolting means sudden increasing of the plant stem before its flowering. That is called as the bolting. In generally in rosette plants like the cabbage, that means in biennial plants, they show the rosette habitat. For example, cabbage, the plant sudden increase its height prior to the flowering. It is called as the bolting. Next, flowering. Flowering, generally gibberellins promote the flowering in long day plants. That exogenous application of gibberellins promote the flowering in long day plants. Long day plants means which require the long days. That means on short nights. Long day means daylight is more and night time period is very short. That plants are called long day plants. Generally, long day plants require winter. They has to expose to the winter prior to the flowering. But when the gibberellins are applied exogenously, the long day plants will flower. Next, substitute of vernalization. Low temperature requirement of the plant promotes the flowering response is called as the vernalization. That means particularly some plants like the wheat, rye, barley and other biennial plants, they are exposed to the low temperature prior to the flowering. That is called as the vernalization. That exogenous application of gibberellins substitutes the low temperature requirement in such a plants which require the vernalization. That means it causes the flowering in the plants which require the low temperature. That means it is called as the substitute of vernalization. Next, parthenocarpic fruit development. Parthenocarpic we learned from the Auxinns, the parthenocarpic fruit development. Here also gibberellins also induce the same parthenocarpic fruits in grapes, cucumbers, etc. Next, sex determination. Like the Auxinns, gibberellins also cause the sex determination. That means development of the pistilate flowers in maize but promotes the staminate flowers in maize. That means paradoxical things are also observed in this sex determination. Gibberellins promotes the formation of pistillate flowers in maize. Pistillate flowers means the female flowers, which it is required for the fruit set and seed set. And in case of staminate flowers in maize, spinach, cucumber, hemp, etc. Next, improve the fruit size and shape. The gibberellins are commercially used to increase the stalk length of the grapes, thereby increasing size of the grapes. And in apple, gibberellins improve the fruit size and shape. Commercially, gibberellins are using for the improving the fruit size as well as the shape in the grapes as well as the apples. These are the physiological effects of the gibberellins, and we can observe the bolting phenomenon in the plant. Here it is the dwarf plant in the first figure. In second figure, prior to the flowering, we can observe the elongation of internode. This is called the bolting, and this is the increasing of size of the grapes. In the first figure, there is the very clumsy arrangement of the grapes, but in second figure, you see that size of the fruit is improved because the effect of the gibberellins. Okay, thank you.